Okay.
Okay.
Cool, thanks.
All right, I'd like to meet, like all of you to meet and say hello to Dr. Alison Narayan. Say hi. Hi, Alison. Yes, she holds a PhD in organic chemistry from University of California, Berkeley, where she worked in the lab of Richard Sapong. Richmond Sapong. Okay. And she finished her degree in chemistry from University of Michigan, and then did her PhD, and then came back to the University of Michigan to do her postdoc.
I just love it here in Michigan.
Yeah, I know. She was born and raised in Frankenmuth, the Christmas capital of Michigan in the world.
Yeah.
And you don't want to hear me talk. Let's have you introduce your topic, and welcome to Andrews University.
Thank you so much for the introduction, and Dr. Roy for the invitation to come. This is my first time at Andrews University, and I've been completely impressed from like the sun just like shining down and all the people walking around smiling. Like I could tell this is a really happy place.
Any snow you see here, any snow is all Photoshop. It's like this all the time.
I've been really impressed by all of the students and faculty here and the awesome research that's going on and the innovation in the classroom. So it's been an awesome afternoon so far. And so today I want to share with you a little bit about my path as a scientist, some of the science that my group's doing at the University of Michigan, as well as some of the opportunities at the University of Michigan, since so many of you are students. I might be thinking about science beyond Andrews University. So it's like a quick poll. Who is going to be applying to graduate programs forward from Andrews? I already talked to some of you. I knew there were a few plans. Yeah. Okay. And so the University of Michigan has lots of opportunities, which I'll tell you a little bit about, but also if you have questions, you can always like get in touch. And so I think it's pretty unlikely actually that I'm standing here giving a talk as a chemistry professor, because when I started my academic adventure at the University of Michigan as an 18-year-old, my vision, right, is that my career was going to look like this. I wanted to be a lawyer. I started as a political science major. And by some random sequence of events and just not knowing any better, my first semester of college, I ended up here in this huge class taking organic chemistry. And
no, wait a minute. Okay. All right. So your first year you took organic?
That's how it's done in Michigan.
Oh, that's how it's done in Michigan?
So you can, during orientation, you take all the series of placement exams, and one of them is a science exam. And so if you do well in it, they say, you don't need Gen Chem, just go straight into organic. And since it's a liberal arts education, you need to take a science class. So I said, okay, I placed into organic. I'll take that. I didn't realize that was atypical for a political science major. But I found myself in this classroom, and throughout the course of the semester, I really started to love it. And it was something that I enjoyed thinking about, structures, and thinking about how you could make those molecules and the cool properties of those molecules. And I quickly figured out that I was actually a chemist. And so I got involved in research, and from that point on, there was no going back. And so forward from Michigan, I traveled across the country to Cal Berkeley, where I started my PhD in chemistry. So it was a pretty natural transition, same school colors, keep my same wardrobe. That was easy. And so there, I worked on organic synthesis in the lab of Richmond Sarpong, just like learning the tools of synthetic chemistry. And then as a postdoc, I moved back to Michigan, and I haven't been able to move ever since, because I've just found it to be such a wonderful research community there. And I'll tell you a little bit about more throughout the talk. And so the research that I've done from the point of being a graduate student on has always been in this area of natural products research. So all the molecules on this slide are natural products, which means that they're made by living things. Some of them are made by microbes in the soil. Others are isolated from animals, and we're not sure if the animal is actually making it or something that lives inside that animal is actually making it. A lot of natural products are isolated from plants and trees. And what originally attracted me to these types of molecules is the structural complexity, right? These things, so many stereocenters and rings. Think about how you could possibly build these is amazing. But what's really kept me in this area of natural products research is the actual properties of these molecules. over 50% of all cancer drugs are actual structures derived from natural products, like halocondrin E. Antibiotics are another class of molecules which are very frequently natural products. So this complex molecule here is vancomycin, an antibiotic that's currently used in the clinic. And there are, natural products come in all shapes and sizes. There's this little one called tetrodotoxin that's isolated from the Japanese fugu fish, as well as these rough-skinned newts that are native here in North America. And these molecules are very potent binders of voltage-gated sodium ion channels, which makes them extremely toxic. And so these animals use this as a defense mechanism. And so it's really two different approaches to natural products that I've studied over the years. The first, the world of synthetic chemistry, where we look at a natural product and think about how could I make that in a lab. And so as a chemist, you start to peel this thing apart to simplify it through bond disconnections until you get back to something that you think that you can start to build from commercially available materials. And even then, sometimes you have to invent new reactions in order to execute that strategy and build the natural product. From the perspective of biosynthesis, we also start at the natural product, but then start thinking about questions like, what makes that natural product? In the case of this molecule saxitoxin, answer is different strains of freshwater cyanobacteria, as well as dinoflagellates that float around in the sea. And then other questions are, what are the genes that encode for the production of that molecule? And if we can learn about the proteins that these genes encode for and their function, we can really start to understand how, at the chemical level, these natural products are stitched together. And so it's really this area, biocatalysis, in understanding the reactions that are mediated by these enzymes in the context of natural product synthesis, is where my group does a lot of work. And we think about these reactions, and if we can develop these methods in a way that's general enough that they can really cross over into this realm of mainstream synthetic organic chemistry, and this would be like a way that people build molecules. So just a quick poll, how many people in the room have taken organic chemistry lab? A lot, okay. You're unsure. Not willing to commit to that, that's okay. I feel that way sometimes too. And so how many of you have done actual reactions in the lab, not just like TLC and things like that? You've run reactions. Okay, and how many of you have run an enzymatic reaction in the lab? Oh.
Well, you mean in an organic lab?
Yeah, in an organic lab. For the purposes of building a molecule. Yeah, so there are examples.
Yeah, we run, but not for biosynthetic reasons.
So there are enzymes that have been used in organic chemistry for a long time. And so some of the best examples of these actually come from industry, people that need to efficiently make a lot of specific compounds. And some of the types of reactions you'll see people using biocatalysis for in industry are simple functional group interconversions, like the hydrolysis of this nitrile to an amide, or to make small chiral building blocks. Enzymes are great at controlling stereochemistry, so you might use an enzyme to make these types of molecules, which then can be a starting point for a further chemical synthesis. So more recently, we've started to see enzymes applied to the synthesis of more complex molecules. And so this is one of my favorite examples. It comes from Merck's synthesis of the type 2 diabetes drug Januvia. And here they engineered this enzyme called the transaminase to install this chiral amino group at the last stage in the synthesis of this molecule. And so some of the things that are great about biocatalysis is that the enzyme provides an environment to do chemistry that can be highly selective. So you can just select one group to do a reaction on in that ring. It's scalable. We can do these reactions safely and in very large batches or in flow. An enzyme provides you with an opportunity to tinker with that and make mutants of your enzyme, which allows you to really tune the catalyst to have the exact properties that you want it to have. But one of the most important properties of biocatalytic reactions is that they tend to have an excellent sustainability profile and safety profile, which makes them a great choice for industry. Okay, so that was the hard sell on biocatalysis. And so with all those positive attributes for biocatalysis, now I'm gonna surprise you by telling you that we're really here. We're like in the Oregon Trail days of biocatalysis. We don't have the breadth of reactions in biocatalysis that you typically have during traditional organic synthesis. And so really it's the mission of my research group and others working in this area is to really move us into the 21st century and develop more sophisticated tools in this area. And so when we think about sophisticated tools, complexity generating reactions that are mediated enzymatically, of course, my history with natural products takes me right back to this place where we think about the fact that nature is really the best chemist, right? And has evolved tools to build these extraordinarily complex molecules. And it's really the perfect time to do this type of research because of technology advances that have happened in sequencing technology and in bioinformatics. And so we're living in an age where if you know there's a certain strain of cyanobacteria that produces this natural product and a toxin, you can sequence that strain of cyanobacteria and then use bioinformatic tools, computer programs that basically will stitch together all that DNA and allow you to search for enzymes that look like they might produce that and identify the gene cluster. And so the gene cluster is really all encodes for the production of all the enzymes that are responsible for the chemistry that puts together this structure. And so we look to these gene clusters to discover new catalysts and think about the types of chemistry we would be able to do with those catalysts. In some of the particular areas that we're interested in, the types of reactions we're interested in, are things like a CH oxygenation reaction, where in a complex molecule where there are tons of CH bonds, right, we always forget about them, we don't draw them in, but they're still there, right? On this molecule, we have all these CH bonds, and there are enzymes that have evolved to select a specific CH bond and transform that into a CO bond, right, to install this hydroxyl group. So we're interested in identifying these catalysts, learning how they work, and applying them in synthesis. We're also interested in enzymes that can take simple building blocks and control how they come together in space. And so we're interested in these types of transformations where you'd bring together two flat molecules and control the stereoselectivity and the site-selectivity in those reactions. Okay. So once we identify an enzyme or a class of enzymes that we think do a really interesting reaction, like what do we do from there? So our goals are to characterize the reaction that the enzyme does in its natural product biosynthetic pathway. So we want to know what it evolves to do. And so one of the projects that we're working on in my lab looks at the chemistry that's involved in the late stage hydroxylation. So taking this pre-saxitoxin molecule, so it's a precursor to the natural product. And there are a series of enzymes that are responsible for selectively hydroxylating all over this core. So I have a student who's looking at each of those enzymes and understanding which CH bond they select for. Once we know what reaction an enzyme does, then we're interested in saying like, okay, we know what it does in the biosynthetic pathway that it evolved as a part of, but we want to know what other substrates it will operate on, right? You can go into the lab and pull things off the shelf and just see what will it do. You can synthesize specific substrates that might be a little bit closer structurally to that natural substrate. And once we kind of hit the limit of what it will do naturally, we want to think about how we can engineer that catalyst, engineer that protein in order to go beyond what its natural substrate scope is. And then, because I'm a synthetic chemist that likes building molecules, we want to see how we can actually use these as tools in synthesis, in multi-step synthesis, to efficiently access molecules that could be interesting to study the biological activity of. Okay, so let's check in on time. We're still pretty good, we still have time. So the specific chemistry that I want to tell you about today is this oxidative dearomatization of a phenol. All right, so a phenol got an aromatic ring with hydroxyl group on it. That's the basic functional group that we're working with. And so this reaction in oxidative dearomatization, so we're installing a new hydroxyl group, which is highlighted in red, And we're also setting a stereocenter in that process. And then we've also taken our aromatic ring and we've de-aromatized that now, right? We no longer have that cyclic array of p orbitals, and so we've created a reactive intermediate that we can do further chemistry on. And so this was a transformation that we were interested in because there are chemical ways to do this transformation, but usually the way that this is done is using stoichiometric reagents. So stoichiometric amounts of copper, hypervalent iodide, or lead. And even with these reagents, there's still a lot of trouble in this reaction to control the site selectivity. So on this ring, both of the ortho positions as well as the para positions are activated to undergo an oxidation, right? So if you want to just react at one of those three positions, it's challenging. Further, if you want to do this in a stereoselective way, you have to control. So we've got our phenol, it's like a flat pancake. If you want to control the configuration of the stereocenter you're going to set, you need to control whether that hydroxyl group is being added from this side or that hydroxyl group is being added from this side of the molecule. And so with these types of reagents, it's a big challenge. And so people have developed these chiral reagents, but they have to use them in a stoichiometric way, right? And that's pretty wasteful. So we started to think about how nature accomplishes this reaction, and it's actually quite efficient and beautiful. And so we got drawn into this area. So nature's tool for doing this kind of reaction starts with an enzyme, a protein that has this flavin cofactor buried inside, bound inside. So that cofactor gets reduced by a molecule of NADPH to the reduced form FADH2. So that reduced form of the cofactor can then interact with a molecule of molecular oxygen. So that's our stoichiometric oxidant in this case. You guys like this?
No, I just recognize the G6PDH.
Oh yeah, it's a recycling system for NADPH.
That's correct. It's
Yeah.
the pentose phosphate pathway.
Boom.
Boom. Boom.
You guys know it?
Boom.
You guys know it?
Boom.
It's really useful. It's really useful.
It's very useful.
Yeah. So otherwise, if we didn't have that system, we would need to use a stoichiometric amount of NADPH, which is really expensive. So instead of doing that, we just recycle NADP+. Okay, so we get to the reduced flavin molecule, and then it can react with molecular oxygen in order to get to our oxidant that we're going to use in our reaction. And so here, this hydroperoxyflavin molecule, those two O's, we've got a peroxide here. This terminal oxygen with the hydrogen attached is electrophilic. So you can think of this as having a partial positive charge on it. I don't know, for all the organic people, if you've ever used MCPBA in order to do epoxidations. I like to think of this as like chiral MCPBA. It's like bound within the active site of the enzyme, but it performs the same type of chemistry, it has the same type of reactivity. Okay, so at this point, at this stage in the catalytic cycle, a molecule of our substrate that's bound within the active site of the enzyme can approach, and this electron-rich substrate can attack that electron-deficient, that electrophilic oxygen, which then gets transferred to the product molecule. Okay, so the challenge in this reaction, like I talked about in the previous slide, is the selectivity, right? So we need to control, so we've got like, my thumb is like the peroxide group. We have to control what face of the molecule is going to approach that, right? Those will give us the two different stereochemical outcomes. And then also the orientation of that substrate, which is going to control which carbon actually gets hydroxylated. And so an enzyme is an excellent tool in this reaction because you can think about if you have this hydroperoxyflavin bound and your substrate bound, we've got the chiral pocket of the active site and all those residues lining the active site that are going to perfectly position that substrate the same way every time in order to get selectivity in our reaction. Okay, so this is great. And then we needed to start somewhere, actually start somewhere in the lab, right? This is all just like what's in the literature. So next, we went to see what kind of enzymes are out there that do this chemistry. And there are sequences for many things. Only a few, those boxed in red, people have actually looked at the reactions that these do in the context of the natural product pathway. And so we wanted to use these as a starting point. So we started with these three enzymes, TROP-B, AZH, and SORG-C. And in the lab, we synthesized what we thought to be the natural starting materials for these enzymes. And so they're all these really electron-rich resorcinol compounds. So when you have two OHs on a benzene ring in this 1,3 orientation, those are called resorcinols. And so these resorcinols are very electron-rich and can undergo this reaction catalyzed by different enzymes. One thing that attracted us to these three enzymes is that you have different site selectivity. So the first two enzymes always hydroxylate between those two oxygen substituents, whereas the third always goes to a different position. So we thought this was interesting to start to look at. And then these are all just intermediates in the natural product biosynthetic pathway, which are elaborated by other enzymes into these different natural product cores. Okay, so what do we actually do in the lab? So I know I talked to some biochemistry majors, so for you guys, this is probably very familiar. We start with a plasmid that contains the gene for the protein that we're interested in. We transform that into a lab strain of E. coli, and we use the E. coli to heterologously express our protein. So what we're left with at the end is an E. coli cell that's like 90% our protein. And then we can bust those cells open, lyse them open, and run a nickel affinity column in order to isolate fairly cleanly our protein. From here, we test the activity in these little 50 microliter reactions. Here, the product is bright yellow, so when the reaction turns bright yellow, it's like a good day. My students are going to go, yes, we have active protein, yay. And so that happened when we got our hands, we synthesized these substrates, and we got all three of these enzymes expressed and purified. And then one of the ways we measure the activity is to use a total turnover number. And so that is how many molecules of product do you make for every molecule of your catalyst. So here, 1,000 represents complete consumption of the starting material. And so then the next question we had is, well, these enzymes are active. They work on the substrates that they evolved to work on. But as chemists, we want to be able to give them all different kinds of substrates. And so we need to learn about what they will do this oxidative dearomatization on. And so my first graduate student, her name is Summer, and she's a synthetic organic chemist. She set out to make as many of these types of compounds as she could. So with this resorcinol type pattern and varying the substituents at all these positions, as well as monophenol compounds, which are less electron rich. We didn't know if these were going to work. And so she made about 50 of these compounds and started screening the activity. And so here, the structures aren't on this page, but we'll get there. So here, just the substrates numbered, looking at the activity with each of those three enzymes. So here again, 1,000 represents complete consumption of starting material. 0 means no reactions happening. And our big takeaway from this is you can start to learn what types of substrates each enzyme could process. Another way to look at this data is to see that in this like Venn diagram style representation is to see that each enzyme really has a different subset of substrates that it likes. And so at this point, we had all of this data, and we were pretty sure they were making products. They all were turning yellow. But as chemists, we wanted to be able to scale these reactions up, isolate the material, hold it in our hand, take NMRs, rigorously characterize this, and see what's going on. And so one of the things that was unique about these enzymes is that they have different site selectivity. So TROP-B and AZA-H both like to hydroxylate at that position, whereas SORB-C, the third enzyme, hydroxylates in an orthogonal position. And so one thing that we found is if we gave TROP-B, for example, a substrate that doesn't have a substituent there, it still hydroxylates at that position. So like every substrate you give it, it only hydroxylates at that position. And so if you have a substituent here, like a methyl group, you'll dearomatize and you get a dearomatized product. But if you don't have a non-hydrogen substituent there, what happens is you can just deprotonate to rearomatize and you just get a hydroxylation of your ring. But the site selectivity is maintained. And we see the same thing happen with SORB-C. All right, and so the next thing we wanted to look at was stereoselectivity. So this is like what makes enzymes great, right? They can completely control site selectivity and stereoselectivity. And so here, we first needed to figure out a way to make a racemic product. So we can do that using a hypervalent iodide reagent, IVX. And we can separate that using a chiral column on an SFC. Anybody use SFC? Supercritical fluid. So basically, instead of using a separation system where you have two different solvents, like acetonitrile and water and an HPLC, you have one solvent, like methanol, mixed in with a supercritical fluid, usually CO2. And so you can do these types of separations. So we developed a method to separate the RNS enantiomers to show that we can separate those, and then compare that to the material that we make enzymatically to show that we're only making one of those enantiomers.
Are you running that on a normal HPLC system?
No, it's like a, yeah. Yeah, so we, in the chemistry department, in the core, like in some of the core facilities, they have an SFC that has like 10 different columns on it, so like different chiral columns, and then they also have a prep column on it, so you can actually do preparative separations this way, which is a really nice tool, because sometimes if you just can't quite get a separation using HPLC, like SFC, it's just different enough.
Dr. Albert makes it up.
Yeah, for certain types
She's trying
of...
to separate chiral things, I think.
So we actually struggled for a long time trying to separate this on a chiral column on our HPLC. We went through like several columns and like so many conditions, and we finally developed a method that like, it's like at 40 minutes, like start to see the peaks come apart. And then we went over to the SFC, and I don't know if you can see, oh, actually this is from the HPLC. So you can see these retention times are like 30 minutes, 40 minutes, terrible. But when we switched over to using the SFC, now, like my student sent me data and I was like, wow, you got the time down to 15 minutes. And she was like, no, that's 1.5 minutes. Yeah, like super fast separations. It's an amazing, amazing tool. We're really thankful for that. So as we started looking at many different substrates, unnatural substrates, so for Trop B, that's its natural product that it makes, and we can change the structure to make different types of products, and the site selectivity and the stereoselectivity is maintained. The same is true for AZA-H. It works on a whole variety of things, including things that are more electron deficient, which are a particularly challenging set of substrates. Sorb-C, the third enzyme that gives this orthogonal site selectivity, doesn't have as great a substrate scope. It has this like long greasy chain, which seems very important to the way that it binds. Without this long greasy chain, we don't really see any reaction with those substrates. And so we're working on trying to understand a little bit about why that is the case. And one of the ways we think about this is just going back to the sequence for each of these proteins. And so we know a lot about the reactions that they'll do, but then we want to think about like what's actually different in these sequences. And for the most part, the sequences are fairly conserved, and there are only a few differences in the active site where you see major differences in the types of residues there. And so we're trying to understand what exactly controls the different selectivity and substrate scope of each of these enzymes. So thinking about just the sequence of the proteins is like one way that we can start to do this analysis, but then this is really further facilitated by having a structure. So then we can really have a 3D picture of exactly what the active site of our protein looks like, and gives us a way to start making more robust hypotheses about this kind of thing. And so one of the things we're working on is identifying residues that might be providing a lot of steric bulk in the active site, and maybe are a reason why some of these substrates don't fit very well in our active site. And so we mutate these residues that are highlighted in green for substrates like this one with Trop B. When you start to make the substituent larger off of the ketone, we start to see the reaction yields go down and down and down. But if we make some of these residues smaller, we can start to see that yields climb back up. And so even for some substrates where with the wild type Trop B we see no reaction, we can make mutations, for example, to these tryptophan residues and now start to see a little bit of product forming. Okay, so I want to wrap things up. I know we're kind of like at the end of our time with a few more minutes, but the last goal is really to think about how we can apply this in complex molecule synthesis. And so the first challenge that we need to tackle is really going from these like tiny little 50 microliter reactions, right, to think about how do you do these on a gram scale? Because we can express protein and purify it and get enough to do smaller reactions, but if you really want to use this and scale it, we need to think about how we're going to do that. So part of our solution was actually to switch the platform for how we were handling the enzyme. So instead of doing an expression E. coli, lysing open the cells, purifying that protein, and for any of you who have done a protein purification, you might appreciate that it takes a lot of time. Sometimes your protein starts to lose activity. Maybe your protein is thermally unstable and you have to like do it in a cold room that's like 4 degrees. All of this is not fun, right? It's labor intensive, it's hard. And so our solution was to just take those E. coli cells that were like 90% our protein and use them directly in the reaction, just use those whole cells. And so our chemistry worked that way. And so that was great for a while until the chemists in my group said, that's okay, but like we really don't like handling goopy E. coli cells. They're gross. So we took it a step. We took it a step further, and now our current process is we take those E. coli cells and we lyophilize them, we freeze dry them, so we just get like a powder of these cells that contain our protein. And so we just throw that freeze-dried cell powder into our reaction. We can weigh it out like any other reagent you would use, and then we still get that nice activity and we're making our bright yellow product.
With the whole cells?
So what we do is we add like usually like an organic co-solvent, and that can help permeabilize the cell. And so you can just get efficient substrate in, product out. It is somewhat dependent on the properties, like the solubility properties, and like how polar or non-polar your molecule is. So there have been a few that have been challenging, but for the most part, it's a general strategy. And so at this point, we're like, okay, we can scale up these reactions. Now let's think about how we can do chemistry with them. And our idea is that we wanted to start from these resorcinol molecules, which we could put together just in a few synthetic steps in the lab, and then think about cascade reactions we could do using either multiple enzymes in one reaction flask or an enzymatic step coupled with a chemical step in order to rapidly build up this complexity. And so here's an example of a multi-enzyme cascade that we're using. And one of the reasons that enzymes are so great for synthesis is that you can do this. You can just couple together all the steps. Like imagine if you're like doing a synthesis and one step is acid catalyzed, and then the next step you're using a Grignard reagent. Like you can't put those together in the same flask. No, you can't. You got to work it up. You got to do the next step. But one of the great things about enzymes is they all basically have the same optimal conditions, right? Aqueous buffer, about neutral pH, and their synthetic machinery, right? Their catalyst is like buried inside their active site and it's protected from everything else that's going around them. So you can drop multiple catalysts into the same flask and they usually play well together. So that's the case with this 2-enzyme cascade that we're using in order to make these types of tropolone natural products, which we're interested in because they have a diversity of biological activities. And so here we can do this initial oxidative dearomatization with trope B, and the second enzyme, trope C, can do a hydroxylation of this methyl group in order to get to this diol, and then it undergoes a ring expansion, a pinnacle type rearrangement in order to make the tropolone product. And so trope C is actually another really interesting enzyme, and it's initially the enzyme we wanted to study. This is what got us into this whole project, but we found that it was so challenging to make these dearomatized products that we wanted to come up with an enzymatic way in order to accomplish that. And so trope C is this non-heme iron enzyme, which really does this beautiful CH hydroxylation reaction where you extract a hydrogen atom to generate this carbon-centered radical, which undergoes a rebound mechanism in order to get to this hydroxylated product and then undergoes the ring expansion. So here we start off with the starting material, and then the tropolone is kind of like an orangey brown color, so we still have the nice colorimetric metric to use here. We can also use this coupled with a chemical step. So here the initial oxidative dearomatization gets us to this 6-membered ring, which has this nice diene embedded in it. So you can throw in a dienophile and do this 4+2 cycle addition. It deals all the reaction in order to build this 2,2,2 bicycle. And so we're working on applying this chemistry to different classes of natural products, including these molecules like the Azaphilones, which here we need to solve the problem of like the stereochemistry, right? We need to be able to access both enantiomers. And so in order to do that, so far we can only access one enantiomer. We returned to nature, found another enzyme that came from a natural product pathway that should have the opposite stereochemistry, and then getting that up and running in the lab, we can see that before we were only able to make this enantiomer, but now with the new enzyme A4D, we're able to access those enantiomeric products. Okay, so these are our main missions, right? We want to figure out what the enzymes do naturally. We want to think about what kind of substrate scope they have and how we can start to engineer the proteins to do what go beyond what they do naturally. And then really apply them in synthesis and provide a tool to more efficiently make molecules. We're also thinking about how we can use this chiral MCPBA type cofactor to do other types of chemistry and do new types of reactions this way. Okay, so I'm really lucky that I've been able to work with talented undergrads, master students, PhD students, and postdocs in my lab. We work at the Life Sciences Institute at the University of Michigan, which is an interdisciplinary building. You guys have the advantage of having chemistry and biochemistry and biology like all right here together. And so it's similar to that at the Life Sciences Institute, which is just creates a wonderful environment to do science. So we've got traditional synthetic space, and we also have a biochemistry lab where we're doing cloning and expressing our proteins. And so these are some of my group members. They're a really talented group that have earned lots of fellowships. I'm a part of the chemistry department in Michigan as well as the Life Sciences Institute, and we've benefited from funding from these agencies. And the last thing, in the last just 2 minutes, couple minutes, I want to tell you about 2 minutes. Okay.
No, perfect.
Okay, I want to tell you about like some of the things going on at Michigan, and this is a little bit of a chemistry-centric few slides. But if you have questions about other programs, please let me know. And so you all know what Michigan looks like. Hopefully you know where Ann Arbor is, down over here. Oops, down over here. It's a really great city, so it's listed in all these like top 10 polls. Like you can find us, and depending on what you're interested in. We're a very literate city, great city for raising families, which maybe isn't the first thing on your minds at this point. Most educated cities, best places for recent college grads. It's a really diverse community that brings together people who are motivated and working in all kinds of areas. So we have a lot of notable alumni, but more importantly, we have a lot of notable science alumni in chemistry and also working in other areas. And so specifically at Michigan, we have chemistry programs, including a master's as well as a PhD. And we have a separate, outside of the chemistry department, we have a program in chemical biology that also has a master's program and PhD. And these master's programs can be course-based or research-based. So the chemistry department is pretty large. We have 42 faculty members. The program in chemical biology has over 60 faculty members affiliated with it. And so there's really a research powerhouse. And if you're interested in a specific area of research, you can probably find somebody who's really engaged in that area. We're specifically divided, the faculty are divided into clusters. So for example, I affiliate with the ChemBio cluster in organic, but I also have taken materials, physical, and analytical students. And so we tend to be a very interdisciplinary place. One other thing that I'll point out is we have several faculty that are also working in the area of chemical education. And we have a lot of opportunities for students in that area as well to be developing new curriculum, testing out ideas about how we teach chemistry and changing that. So we have a lot of annual symposiums and relationships with industry. One awesome thing about being a graduate student, unlike going to med school, dental school, or PharmD program, like I talked to a lot of you back there, when you are in a graduate PhD program, we kind of just like take care of you. You get a stipend, you're an employee of you're a student and an employee at the university, and so you just really get to focus on learning and your science, and it's kind of like a really wonderful and special time. So it's mostly a program where you take classes in your first and second year, and then the bulk of the time you spend is actually in the lab doing research, generating new knowledge, and really being at the forefront of science. Okay, so our students are involved in all kinds of programs and get to travel and do all kinds of wonderful things. Some people go on to work at companies, and we have companies that come and do on-campus recruiting, and so our students are really great, and so companies want to come hire them. So a lot of people go to industry, and others go on to tenure-track positions at institutions all over the country, or working in government labs and teaching in other places. So those are some of our faculty. So I'm one of our assistant professors, but we have like a whole bunch of other new people that are working in exciting areas along with me. So Professor Schultz is working in chemistry education, and Frank is really working at the interface of chemical biology and biophysics. Jennifer Brito-Rab is our newest hire. She's doing inorganic chemical biology type of research, looking at some interesting proteins and how they function, as well as Sarah Keene and Kristen Kutmo, who are both doing research related to RNA. And so it's an exciting place to be. We want to see the people joining us who are the best and brightest, and just from being here for the afternoon, I already get the sense that these people, you are the ones we want. So if you have any questions, definitely let me know. I guess I'll also mention that we have opportunities for summer research. So while you're still involved in undergraduate education, to come and have a research experience for the summer through many different programs. So if that's something you're interested in, let me know. There are probably like 5 or 6 different programs you could apply to at Michigan. Yeah.